Fakultas Kedokteran Universitas Airlangga, berlokasi di Kampus A Universitas Airlangga, Jalan Profesor Dr. Mustopo, nomor 47, Surabaya.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Fakultas Kedokteran Universitas Airlangga merupakan salah satu fakultas kedokteran terbaik di Indonesia yang telah berusia lebih dari 1 abad. Dan kita mempunyai visi dan misi Fakultas Kedokteran Universitas Airlangga saat ini merupakan fakultas kedokteran yang mandiri, inovatif, terkemuka di tingkat nasional dan internasional, serta pelopor pengembangan ilmu pengetahuan dan teknologi kedokteran, entrepreneurship, serta humaniora berdasarkan moral agama, excellent with morality. Salah satu divisi departemen di Fakultas Kedokteran adalah biokimia kedokteran, yang merupakan salah satu departemen preklinik yang banyak memberikan sumbangan keilmuan dasar kedokteran dan dapat terlihat dari produktivitas dengan karya-karya sindikat. Baik itu untuk pendidikan, pengajaran, penelitian maupun pengabdian masyarakat dan publikasi yang bertaraf nasional internasional. Tahun 2020, seluruh stafnya untuk Departemen Biokimia sudah meraih gelar doktor dan beberapa menjadi guru besar, serta mempunyai calon-calon penulis artikel yang luar biasa berkualitas tinggi dan mendapatkan penghargaan manuskrip saat ini. Departemen Ilmu Faal dan Biokimia Fakultas Kedokteran Indah menjadi pelopor dilaksanakannya program pengabdian kepada masyarakat berskala internasional dengan melibatkan kolaborasi antara universitas dalam negeri maupun luar negeri. Demikian, salam sejahtera dan sehat selalu untuk kita semua, stay healthy dan punya hebat. Assalamualaikum warahmatullahi wabarakatuh.
Fakultas Kedokteran Universitas Airlangga terdiri dari 29 departemen. Salah satu di antaranya adalah Departemen Ilmu Faal dan Biokimia Kedokteran. Departemen ini terdiri dari 2 divisi, yaitu Divisi Ilmu Faal dan Biokimia Kedokteran yang semula terdiri dari 2 departemen yang merger pada awal tahun 2021.
Selamat pagi dan salam sejahtera bagi kita semua. Departemen Ilmu Faal dan Biokimia Kedokteran merupakan salah satu dari 29 departemen yang ada di Fakultas Kedokteran Universitas Indah. Biokimia Kedokteran sebagai ilmu dasar memiliki peranan yang sangat penting bagi perkembangan ilmu sepanjang kehidupan manusia yang mampu menjawab berbagai persoalan dan tantangan dunia. Karena seluruh kehidupan di alam semesta tidak bisa lepas dari interaksi molekuler. Sebagai ilmu dasar, biokimia terutama berkaitan erat dengan ilmu kesehatan. Di samping itu juga di bidang pertanian maupun industri. Terkait visi dan misi Fakultas Kedokteran Universitas Airlangga untuk menjadi fakultas kedokteran yang bermartabat, kompetitif serta unggul di tingkat nasional dan internasional, berbasis riset dan teknologi terkini dalam menunjang proses pendidikan dan pengabdian masyarakat, maka kegiatan di Departemen Biokimia selalu mengacu dan mendukung Tridharma perguruan tinggi yang telah direalisasikan dalam bentuk menyelenggarakan kegiatan pendidikan di seluruh jenjang pendidikan mulai dari S1, S2, dan S3. Hal ini didukung oleh para staf pengajarnya yang selalu mengikuti perkembangan ilmu biokimia dan biologi kedokteran yang mencakup omics serta melakukan pengembangan dan penguatan tele-education. Samping itu juga dengan melaksanakan penelitian di tingkat nasional dan internasional serta pengembangan ilmu biokimia dengan pengembangan teleseminar atau pelatihan biologi molekuler mencakup omics serta pengembangan omics dalam rangka pemantapan biologi molekuler kedokteran pada pendidikan profesi dan pendidikan spesialis. Juga dengan melaksanakan kegiatan pengabdian masyarakat yang berkaitan dengan pemanfaatan ilmu biokimia kedokteran dalam kesehatan dan kesejahteraan masyarakat yang berbasis riset, bersifat kolaboratif dan berorientasi pada pemberdayaan masyarakat dengan menggalang dan memperkuat kerjasama di tingkat nasional maupun internasional. Demikian gambaran divisi biokimia kedokteran.
Good afternoon, Prof. Indah. Mbak Lia mungkin bisa di stop dulu ya.
Good afternoon and Assalamualaikum warahmatullahi wabarakatuh. Salam Ramadan.
Warahmatullahi wabarakatuh. Salam Ramadan, Prof. Indah. Thank you very much, Prof. Indah. Maybe we still have time here. Perhaps you can check on the performance first. Maybe you want to try to share your screen to check on your presentation, Prof.
Yes, so now I'm sharing my slide. Great. So many things on the desktop. I will close now. Wait, so can you see the slide? My slide? I'm sharing the slide right now.
Yes, we can see the slide.
Okay.
Perhaps you can try to slideshow
Okay.
and try to move it, please.
Okay. Is it okay? Now
Yes, it is okay.
Okay.
Yes, thank you very much, Prof.
So do I need to stop share now?
Yes, please stop the screen. We still have about 12 minutes here.
Okay.
Welcome to our department, Prof. We
Yeah.
are from the Department of Physiology and Medical Biochemistry.
Okay.
And
So,
the
yeah.
And Dr. Gwenny here is the head of our department, also Professor Retno, Professor Sucipto, Dr. Emma, Dr. Anissa.
Okay.
We're glad to have you as our collaborator here.
Okay. Assalamualaikum to all.
Waalaikumsalam warahmatullahi wabarakatuh, Prof.
Okay. How's the Ramadan there?
Alhamdulillah. Everything's fine. And how are you, Prof. Indah?
Alhamdulillah. Right now we are having a face-to-face session, meaning that no more online. Okay, we are calling all our students to go to their formal classroom. So we are having a physical class right now.
Oh, I think it's the same with us, right, Dr. Emma, Dr. Gwenny, and the others.
Yes, yes. We are now also having an offline class.
And also, I cannot call my student because right now they are all in class. They got class now. So That's why I'm not calling them to attend this lecture. They have another class with another lecture.
It's fine. Thank you, Prof.
Okay. So before we start, can you all maybe a brief introduction about yourself, about what you are doing, especially in research?
Okay, maybe from Dr. Kweni as the head of our
Maybe.
department. Dr. Kweni, please.
In our department, about the molecular genetics experiment and herbal.
Okay.
Maybe Dr. Nisa can explain about herbal alang-alang, maybe.
Yes, Dr. Nisa is an expert in herbal medicine of our department.
Okay.
Assalamualaikum, Prof. I'm Anissa from Department of Physiology and Medical Biochemistry. I'm interested in herbal medicine, especially in metabolic disease and neuroscience. In my dissertation about herbal medicine and memory lack, maybe associated with Alzheimer, but I'm focused in memory deficit. And also, I'm interested to simulation herbal medicine with in silico. In silico, I think, yeah. You know about in silico, docking molecular.
Yeah. I heard that.
Yes, it's my interesting herbal medicine in silico and about the related disease, including metabolic disease and neuroscience. Maybe we can join research or further.
Yes, we can collaborate more in the future, I guess.
Yes. Later when I giving my not lecture, I was mention lecture because I was presenting to my student. Later when I present my talk, okay, you will see what are the plan that we are research on. Okay, so maybe you are having an interest on certain or particular plan. Maybe we can talk later and discuss about our future collaboration.
Yes, it will be interesting. We have a lot of expertise here, and I myself is actually not an expert in herbal medicine, but we together had performed a research about habatus sauda, Nigella sativa.
Oh, I see.
For neural protection.
Oh,
so I guess we can also collaborate more in the future about this.
Okay. Because I came from Faculty of Dentistry, so most of my work is related to oral health, related to the oral cavity. Because I'm in Faculty Dentistry, of course, we need to do something related to our field here. So you can see, but I'm actually my field is about cancer. So if you're already interested to do something in cancer, maybe we can also collaborate.
Yes, Prof, of course, it will be a great pleasure for us.
So who will be attending this talk?
Besides of the lecturers, there are also students from undergraduate, postgraduate, yes, undergraduate, graduate, and postgraduate, I guess. And not only from the Faculty of Medicine, but also from other faculty.
Okay, I see. So, student of medicine there will take this medicine course for 5 years or 6. How many years there?
5 years, Dr. Emma?
Oh, 5
Five
years.
and a half.
5 years.
5 years.
5.5.
Yes, 5.5.
How about the houseman course? Houseman, do you have course like here? Houseman, we call it houseman. After finish their study, they go to the government hospital to work there for 2 years.
Yes, we call it internship.
Internship, yes.
Yes.
We have a
Not
program.
only, yeah, but not only for hospital.
Oh, call it
Government hospital. I'm sorry.
Okay, see.
Yes, they usually apply to medical facilities in Indonesia, and it's not only apply in a government hospital, but also
Oh.
private hospital is allowed.
Oh, okay. You have you are physiology and biochemistry is in one department.
We have
Yes.
two divisions.
Yeah, the division, yes.
Yes, the division of biochemistry and the division of physiology. But the host here right now is the division of biochemistry. So we are from the biochemistry division from that department.
Oh, I see.
Even though there are also some lecturer from the physiology division here, like Dr. Arum, Dr. Lily, and the others.
Do you have expatriate day from other countries that teach your student? Expatriate.
I think it is not, yes.
Not
Dr. Emma.
regularly, yes. Not regularly, yes. But
Not regularly, but
yeah.
sometimes we have such program like this, international academic lecture like this, and most of them come from Malaysia.
Oh, you have Malaysian lecturers there.
Not regularly, just like you apply to the program and we have a guest lecture here like that, but not regular lecture.
Oh, I see. How about the international student?
There are some international students like from Malaysia, Palestine, and from other countries, but not too much, I guess.
Oh,
And what about in your university?
in your ITM, because this is actually a university meant for Malay, for Bumiputra. We call it Bumiputra Malays. Malays, and also the origin of the race in Malaysia. So we only have this student. We don't mix with the international student.
In our faculty, there are two types of class, the regular class and international class. The students from abroad can attend the international class. So there are different classes here.
I think almost 2 years.
Yes, it's almost time. Just 1 more minute. We already have 32 participants here.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Assalamualaikum wabarakatuh. Greetings, Dr. Lily. Dr. Lily is from Department of Physiology, Division of Physiology in our department. Yes, Physiology Division.
I have ever been to a UTM.
Oh.
Yes, I was visiting sport faculty about sports, like something like that.
Oh, I see. Meaning that you are in UITM Shah Alam?
Yes.
Oh, okay. Because dentistry and medicine is at Sungai Buloh campus.
Yeah, it's different.
It's not in main campus.
It's already 1 p.m. Maybe Dr. Emma can start.
Okay, we can start. Can we start now?
Yeah, of course.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, Professor Indah, professors of medical biochemistry and physiology. Ladies and gentlemen, welcome to International Academic Lecture in Physiology and Medical Biochemistry Department. This webinar is organized by Faculty of Medicine, Universitas Airlangga, in collaboration with Division of Medical Biochemistry, Department of Physiology and Medical Biochemistry, Faculty of Medicine, Universitas Airlangga. My name is Emma Kurnianingsih, and I'm very delighted to have you, Professor, doctors, colleagues, ladies and gentlemen, in today's lecture. Today we will discuss about herbal medicine in apoptosis and cancer. And before we start our lecture, please kindly see the agenda for today. So we will have the lecture, the herbal medicine in apoptosis and cancer. And this lecture will be delivered by Associate Professor Indah binti Amin from Department of Preclinical Sciences, Faculty of Dentistry, Universiti Teknologi Malaysia. And we will have also discussion that will led by Dr. Citrawati Dia Kencono, Dr. Magister of Science. And before we jump to the main agenda, the lecture, please allow me to read the curriculum vitae of Dr. Citra as our moderator for today. Dr. Citrawati Dia Kencono, Dr. Magister Science, is a member of Department of Physiology and Medical Biochemistry, Faculty of Medicine, Universitas Airlangga, and she is now currently an active member of Hepatitis Study Group, Institute of Tropical Disease, Universitas Airlangga. Dr. Citra was born in Magetan, December 22, 1988. Dr. Citra Dr. Citra took the education in Universitas Airlangga, the bachelor, the undergraduate program, the magister, and the doctoral program in medical science. And Dr. Citra now is the head of research division for undergraduate, magister, and doctoral program in research unit in medical faculty, Universitas Airlangga, the co-coordinator of journal clinic UPG, medical faculty, Universitas Airlangga, the member of genetic study group, medical of faculty of medicine, Universitas Airlangga, the advisor for Surabaya Neuroscience Club, and the member of clinical research unit in Universitas Airlangga Hospital. And I guess we can go to the main session, and please, Dr. Citra, time is yours.
Okay, thank you, Dr. Emma. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. And thank you for Associate Professor Dr. Indah binti Amin from Universiti Teknologi Malaysia. We are very glad to have you here. And this is the first time Professor Indah gives a lecture in Universitas Airlangga in this International Academic Lecture Program. which is hosted by Department of Physiology and Medical Biochemistry, Faculty of Medicine. She's going to give the lecture about herbal medicine in apoptosis and cancer according to her expertise, as she has conducted many research on this field. As we know, the breakthrough of herbal medicine has reached nanoparticles research and molecular medicine with various applications, including cancer, one of the deadliest diseases which has become burden all over the world. And here Prof. Indah will explain how could we combat this disease through the development and application of herbal medicine. And we're also going to learn about the molecular mechanism. If any of the participants want to give any question to Prof. Indah, you may write it in the chat box along the session or directly ask it by raising hand after the session. Before we start, let me show you the short CV of Prof. Indah. Professor Indah comes from the Department of Preclinical Sciences, Faculty of Dentistry, Universiti Teknologi, Malaysia. And she also has research interests in natural products, cancer research, drug delivery, nanoparticles research, molecular medicine, and herbal medicine. She also has positions as the head of Research Interest Group, Natural Approach to Oral Health Research Group, Entity Kecamatan Tier 5, Universiti Teknologi Mara, and the head of Department Center of Preclinical Science, Faculty of Dentistry, Universiti Teknologi Mara. She also has experience in many organizations such as the Professional Technologies, Malaysia Society for Molecular Biology and Biotechnology, and Malaysian Association of Clinical Biochemists. She has many achievements which I cannot mention one by one, like she has 32 awards, 14 grants, 18 index publications. Okay, I think it's time for the lecture to begin. Professor Indah, the time is yours.
Okay, thank you, the MC. Okay, so I'm sharing my slide. Okay, please allow me to take a second to project my slide. Okay, so can you all see my slide?
Yes, we can, Prof.
Okay, Bismillahirrahmanirrahim. Okay. First and foremost, I would like to thank the Faculty of Medicine, University Erlangga, especially to the Department of Physiology and Medicinal Biochemistry for inviting me for this sharing session in academic lecture. Okay, actually, I'm deeply honored to be here today, as I've read that University Erlangga is the second oldest university in Indonesia. Is it right?
Yes, it's true.
And is consistently ranked highly in major world university ranking. So I've been given the title of herbal medicine in apoptosis and cancer. Actually, I've been involved with the herbal medicine research since 2011, meaning that it's over 10 years already. Okay, since now I'm actively involved in research of herbal medicine, and with my little experience and my small contribution in this field, I would like to attract the audience here to join me, okay, to explore the numerous benefits of our biodiversity heritage, especially in our country, Malaysia and Indonesia. So before I'm going further, okay, this is the outline of my presentation. Okay, this is like a compiling of my research work related to herbal medicine. Okay, actually there are other herbal medicine that is not in our discussion today. This is only a few of the plant that I work with. Okay, first we will discuss about the ficus deltoidea, and then we will discuss about the plant that we call as elocamodin, and then dilapiol, and a brief about the allicin. Okay, what all this thing is actually a scientific name of the plant. Later we will come to the layman term. Okay, later. Okay, so for the introduction, okay, there has been a lot of interest in biological product derived from the mega destination like Malaysia and also Indonesia. In my country, okay, especially in Malaysia, we have a diverse ecosystem and also some of the world's oldest rainforests, making our country as a treasure trove of over 2,000 plants that are reported to have the medicinal potential. And currently in our country also, the pharmaceutical sector has also shifted its attention to the development of herbal goods such as nutraceutical, health supplement, and also traditional remedies. Okay, based on the Dasar Negara, we have here what we call overview of the national traditional and complementary medicine TNC blueprint. This shifted, okay, meaning that our attention to the herbal medicine product could reduce the healthcare cost by 30 million in 2027. Okay, so therefore, several policies and initiatives were taken in our country to promote interest from stakeholders and to push more R&D within the local herbal sectors. So in line with the country goals, our research group in UITM, in my university, especially in my faculty, okay, especially in my group, okay, has took this advantage to focus more on the natural plant product research with our objective to establish ourselves as the leading research group that capable of entering the herbs industrial market. So going on for the first herbal plant, okay, this is what we call ficus deltoidea. Okay, what are the things about the ficus deltoidea? Okay, this is my first study when I involved in the first study that I involved in herbal medicine. Okay, I'm doing in vivo study. Okay, so ficus deltoidea has a long history of usage in Malaysian traditional medicine, and actually it is used, okay, treating ulcer and other diseases. If you can see my slide here, okay, it's a plant, it's a small herb which rarely exceeds 2 meters in height and is domestically cultivated. Okay, in our country, we call it mascote. But I refer to a journal, okay, in Indonesia, you call it tabat barito. I don't know, this is the whether I pronounce it correctly, tabat barito. Do you know the plant? Okay, it's okay. So,
I don't know.
okay. It in our country, it is traditionally consumed as a herbal drink for women after childbirth to help instant strengthening the uterus. Okay, and also it acts as a libido booster for men and women. Okay, so as I mentioned earlier, okay, I'm working in dentistry faculty. So most of my work are related to the oral health development. So what are the common problem in our faculty is ulcer. Okay, oral ulcer occurs, if you can see the picture, okay, I think all of us experience ulcer in our mouth, right? So by definition, oral ulcer is actually mucous membrane in the oral cavity, and it is a solution in the mouth. Okay, so there are many types of oral ulcer. The common one we call it RAS, or the name is recurrent after stomatitis. This is the most common one. It is a painful oral mucosa condition observed in our patient. And if you can see the features of the in the picture shows, it is a small, round or ovoid ulcer with circumscribed margins, yellow or gray floor surrounded by erythematous halos. Okay, in our clinic here, okay, we are used to treat the patient with what we drug that what we call a triamcinolone. Is triamcinolone acetonide, is a dental paste meant to temporarily relieve the symptom of mouth sores resulting from injury. Okay, how it works? It will reduce the swelling, the pain, and ulcer in the mouth. Okay, you can see the result by weeks. Okay, within 1 week you can see the improvement. However, there are side effects which include burning, irritation, dryness, or redness of the treated area. Okay, so what we do in my research group, okay, we do an in vivo study. Okay, we divide this rat, okay, the experimental rat, into 4 groups. Okay, the first group is control group. Okay, the second group, the negative control group. The first group is the negative control group. The second group is the drug that we use to treat the patient. It is a triamcinolone acetonide. And then for the third and fourth group is our experimental experiment. Okay, we use ficus deltoidea, okay, in the concentration of 250 milligram per kilogram aqueous extract, and for the group 4 is 500, a little bit higher. Okay, so okay, this is the result of the experiment. Okay, so you can see, okay, so we are giving the ficus deltoidea 10 days consecutively. Okay, in the day 6 of the treatment. Okay, if you can see my result here. Okay, this is the white color. I hope everybody can see the slide clearly.
Yes, Prof, we can see it clearly.
Okay, thank you. So this white color is actually the positive control. Okay, and then we have our ficus deltoidea, okay, the 250 milligram, and the black color is the 500. Okay, you can see at day 6, okay, the inhibition percentage area of oral ulcer almost shift to full recovery. Okay, and you can see, meaning that our compound ficus deltoidea is better than the positive control. This is the positive control, and we get the result in day 6, and in day 10 we achieve the full recovery. Okay, what is the most interesting one? I mentioned earlier about the triamcinolone. It is give the side effect to the patient. Some of the patient cannot use the triamcinolone. So maybe, okay, based on our finding, actually we can suggest that ficus deltoidea extract can replace, okay, the treatment of oral ulcer. And moreover, okay, in toxicological study done by other researchers shows no toxic effect to the bone marrow, liver, and renal function on rats. Okay, then they are not found any harm to the rats. And other study also prove that ficus deltoidea has no significant toxicity to human. And meaning that Ficus deltoidia leaves across extract is safe for consumption. Okay, so because of this finding, we come up with the new formulation of oral health products from the Ficus deltoidia extract. Okay, we come with a prototype and we bring all our products in this conference in 2016, and we are awarded with silver for this finding. And we managed to publish one paper for this study. Okay, this is the first experience when I'm involved in herbal medicine. And right now I have one student, okay, PhD students are continuing this study, but specifically looking on the wound healing. Okay, move to the second plant, okay, about the aloe-emodin. Okay, aloe-emodin is actually an anthraquinone compound found in aloe vera. Okay, I think everybody know what is aloe vera, isn't it?
Yes, of course, Prof.
Okay, kita in Malaysia we call it lidah buaya. I don't know how you pronounce it in how you call it in Indonesia.
Just the same lidah buaya as well.
Okay. So lidah buaya. Okay. So aloe vera. So this is a main component of aloe vera, but it also found in other species such as Asphodelaceae and Polygonaceae families. Okay. So aloe vera is actually established medicinal plant because many publications come from the aloe vera or aloe-emodin, and they have shown that aloe-emodin have the anti-cancer properties such as anti-migration, anti-invasion, anti-angiogenesis. Okay, this is all the related to anti-cancer activities. And aloe vera is not really new to us. Okay, there are many available publication about the benefits of aloe vera, such as it can treat the constipation, improve the digestion, reduce the gum inflammation, and so on. Okay, so what we are interested on this aloe-emodin. Okay, during that time, okay, when I did the study, okay, we are searching for the anti-cancer effect, and there's so many papers about aloe-emodin that have been studied on many types of cancer, but not really in breast cancer. Okay, so with that, okay, so we find a grant. Okay, so we received the grant, and then we embark on this research that looking on the anti-cancer of aloe-emodin in breast cancer. Okay, and we specifically want to see the apoptosis. Okay, I think I won't discuss more about the apoptosis. We all are here are biochemist lecturer. Okay, just a brief about the apoptosis. Is a program cell death. Okay, it's a natural way when the cells decide to suicide. Okay, how the apoptosis is induced or is stimulated because of certain circumstances such as stress heat, radiation, nutrient deprivation, viral infection, hypoxia, and others. Okay, so we know in apoptosis, if the cell decide to suicide, okay, they will go through a series of mechanism. Okay, series of mechanism. The first in apoptosis, we can see that the cell will shrink. Okay, the chromatin will condense, and then after that, the membrane will start bleeding, and all the organelles inside the cells will disintegrated. But the thing is, what is the most interesting in apoptosis, you can see that the membrane still intact, but it's dish shaped, meaning that the shape is altered. And at the end of the process, okay, it will turn to apoptotic bodies that will be engulfed by the macrophages. So this is the natural way how the cell is die. And what is the most important thing? This natural way will not cause any inflammation. Okay, in contrast with the necrosis, okay, there is many way of how the cells die in our body. For example, necrosis, autophagy, and most of the drug development are targeting on the apoptosis pathway, because as I mentioned just now, it will not cause the inflammation to adjacent cells. Okay, so the natural process of removing undesirable cells or apoptosis actually is disrupted in many cancer. Deregulation of apoptosis lead to the uncontrolled tumor growth and the development of chemotherapy resistance. Okay, so by targeting the apoptotic cell death pathway will open a world of possibilities for drug research and development. So any agent that modulate the apoptosis are extraordinary promise for the treatment of several states of disease, especially in cancer. So this is another view what the difference between necrosis and apoptosis. Okay, so because of these distinctive features, so we have a specific assay to target the to recognize the apoptotic body. Okay, so I've been explain you about the features of necrosis, the features of apoptosis. Okay, so there are very distinctive features of necrosis and apoptosis. Actually, why cancer patients dies? It is not because of the treatment, actually because they are suffering from necrosis, because the toxicity that happened in their cells. Okay, meaning it lead to the necrosis. Okay, this is the explanation of the difference between necrosis and apoptosis in terms of cell size, nucleus, membrane plasma, and also the cellular contents. Okay, you can get it actually in many literatures. Okay, so what I'm trying to focusing here, okay, about the amodine. What is the interesting about this amodine? Okay, this is our control, meaning that the cells without treatment of amodine, and this is the treated cells. Okay, so when we do this experiment is a cellular assay using the dye. Okay, we can see the characteristic of apoptosis features. Okay, EA stand for early apoptosis. Okay, early apoptosis, and this is late apoptosis. Okay, how you can differentiate whether this cells is going to apoptosis mechanism or necrosis? Because you can see the membrane. Okay, see the membrane still intact. Okay. If you compare, because I don't have the necrosis cells here, you can see the cells is like burst, explode. Okay, explode. So this is a sign of apoptosis mechanism, and we also do the flow cytometry. Okay, using another dye, and this also shows that our treated sample has the highest apoptosis percentage, and one of the characteristic of apoptosis happen in our cells is about the DNA fragmentations. Okay, compared to the necrosis. Okay, if your cells is going to necrosis, you can have the DNA, the fragmented DNA. You will have the lyse nucleus, or what we call in biochemistry karyolysis. Karyolysis. So that's why we can measure and prove that our amodine shows the apoptosis mechanism. Okay, so beside the cellular assay, okay, we also do the gene expression and protein expression, and we I will not show here because this is a lot of genes and protein involved. So I just simplified here. So through our findings that shows that our compound amodine induce the apoptosis in both extrinsic and intrinsic pathway by activating the p53 genes or protein. Okay, this is our conclusion. So at the end of our study about amodine, we managed to reveal that our compound amodine from aloe vera selectively inhibit the proliferation of MCF7. Okay, this is estrogen positive breast cancer cells, but not in MCF10A. 10A is a cell line for non-transformed breast cells, meaning non-cancerous cells. And also we also successfully proved that our compound amodine induced cell cycle arrest at G2M phase, which involved the activation of p53 extrinsic and intrinsic apoptosis. And another thing that is also interesting, but because with the limitation of time, I cannot show you the result, but we have successfully proved that amodine synergistically enhance the tamoxifen efficacy. Tamoxifen is a drug used to treat cancer patient. Okay, so what we can conclude from our study here, okay, we are suggesting that amodine could be a potential agent to be used in combined anti-estrogen treatment, and also we are provide strong foundation so the study could be extended to in vivo study, and also widen the possibility to make it to the clinical experiment setting. So this is the papers that managed to publish about the amodine. Okay, if you have time, maybe you can go to the details of the paper. Okay, I cannot explain in this session in this short time. Okay, going to our third herbal plant. This is my student, okay, one of my master students. Okay, she's working on dilapiol, and also working on the finding the anti-cancer agent. Okay, because she registered under Faculty of Dentistry, so we are trying to find research that are related to dentistry. Okay, so we are working on the human nasal squamous cell carcinoma. The model cell line is RPMI 2650 cells. Okay, this is the plant. Okay, the active compound is dilapiol. Okay, so dilapiol is a penyl propanoid extract isolated from Peperomia pellucida. Okay, this is also traditional medicinal herbs. it is reported to possess medical benefits, including antiviral, antifungal, anti-inflammatory, and anti-proliferative properties. Okay, Peperomia pellucidea, in our country, we call it a Siricina. Okay, I don't know you are familiar with this plant. Okay, sometimes we call it kuntumpangan air. Kuntumpangan air. Okay. Okay, beside this medicinal benefit, it also possesses other bioactivities such as cytotoxic, anti-diabetic, antioxidant, antimicrobial, and anti-hypercholesterolemia. Okay, so about the human nasal cancer, this is just to show you the illustration of the anatomical part. Okay, so this is where the cancer is. Okay, what are the treatment? The current treatment for this cancer is surgery, radiotherapy, and also chemotherapy. But all this treatment cause the patient quality life affected. Okay, after the surgery, sometimes the patient cannot really talk because they take up some part of the oral skin or something. Okay, so there is an urgency to find alternative for this kind of cancer. Okay, so this is control one. Okay, without any treatment. Okay, you can see using the microscope. Okay, this is how many times can tak ada tulis kat sini. Okay, this is untreated one. Okay, this is the cisplatin. Okay, this cisplatin is cisplatin is the drug that used to treat the patient. Okay, you can see after 72 hours of incubation, the cells are in the proliferation are inhibited. Okay, this is the positive control, and this is the compound. Okay, the compound that we are using the concentration of IC50 and IC75. Okay, I think you all are familiar with what are the meaning of IC50. Okay, IC50 meaning that how much the drug is needed to induce or to inhibit the half of the biological process of the particular organism. This is about IC50. So using this concentration, you can see the effect of the dilapiol to the oral, to the nasal cancer cell. Okay, so because we are interested to study on the apoptosis pathway, okay, so we do the apoptosis, we use the apoptosis assay, okay, and then we are using a flow cytometer, so we can see in our sample. Okay, this is the untreated one. Okay, untreated without meaning without any treatment. Okay, this is control, and this is our compound dilapiol IC50, and this is dilapiol IC75, and this is the control positive, the drug used to treat the patient, cisplatin. Okay, so meaning that, our compound, both IC50 and IC75 shows a significant percentage of early and late apoptosis. We also do the gene expression and protein expression, but I'm not discussing all the result here. Okay, so as a conclusion, the reduction of the cell viability of RPMI 2650 cells suggests potent cytotoxic activity of dilapiol of our compound at dose-dependent manner, and it also induced the apoptosis as the mechanism for cell death. Okay, through protein experiment, okay, our finding reveals that dilapio activated apoptosis both through the both intrinsic and extrinsic pathway by the upregulation of caspase-8 and BCL2 protein respectively. So we managed also to publish a paper. Actually, there is another paper related to this study. This is the recent one, and I published last year. This is thanks to my master student, Patricia. And last but not least, okay, this is the last herbs. We can't call it herbs, but actually we always eat this apa ni, bawang putih kan? Okay.
Yes.
Okay. So in our faculty, the most common cancers occurs is oral squamous cells carcinoma, and it is contributed 90 to 90% of oral malignancy. So for this kind of cancer, the main treatment is through chemotherapy and surgery. Right now, for chemotherapy, we are using cisplatin. But many reported study shows that cisplatin give us side effects such as nausea, diarrhea, pain, and vomiting. So therefore, there is urgency to find more reliable anti-cancer agent. And one of my students, master student, Farah Hazwani, are working on the lysine. Lysine has been known for decades for its diverse biological properties. What is a lysine? It is an organosulfur compound obtained from garlic. Actually, when we chop the garlic, okay, there is enzyme called apa namanya? Oh lupa. The enzyme that turn iline to a lysine. Okay, that you can smell actually the bau of the bawang putih itu from the lysine. Okay. So despite its function of being antioxidant, we know the garlic is famous with its antioxidant properties, also antimicrobial, antifungal, anti-inflammation, but it also serves as an anti-cancer. And now we are concentrating our work looking on the anti-cancer properties on the oral squamous cell carcinoma. And currently, okay, now my students are doing this study. We successfully shows that our lysine is capable to inhibit the oral squamous cells. But I'm not revealing the result yet because it's not published yet, but we are managed to presenting our study in one of the conference here, and we got diamond. Okay, diamond for this research. Okay, so I think with this limitation of time, okay, I that's all the plan that I want to share. Maybe we can have more time for question and answer. But before that, I would like to share the sweet memory with what we have achieved from 2011 until now. With not only me, okay. I'm not working on this all alone, but with my group, okay, we managed to obtain many awards. Okay, so this is one of the awards. We got diamond. Okay, diamond award, and this is about the lysine for oral cancer. We also got gold award, and this is about the nanonim. Nanonim is I'm not discussing this because this is a research I've done with my co-partner in Faculty of Applied Science. We are focusing on the nanoparticles of nanonim. Okay, this is about the nanonim again. Okay, this is about the what is this? I'm not remember. Okay, this is about ficus diltodia. I did mention just now. And another award from ficus diltodia, we got diamond. Okay, for our project, everyone can smile, no ulcer, no pain. Okay, this is my student and other lecturers that involved in our project. Another award for another compound. Okay, this is a lysine combining with the rose. Okay, so one of the interesting project that we conduct last year, not in 2019, okay, about the garlic. Okay, we managed to show the antimicrobial and antifungal activity, but you see garlic somehow some of us cannot stand off the smell, isn't it? Garlic.
Yes, it's stinky.
So when we first presenting our lysine in this kind of conference, one of the judge has commented about the lysine because how can you give treatment with the smells? So we come out with the innovation. Okay, we combine the lysine with the rose. Rose, you know, the flower. So we come up with the prototype of what we call rose chin. So we got a gold award for this kind of innovation, rose chin, for the product for prototype of oral health product. Okay, so this is the previous. This is the first one. A lysine, the study of a lysine. Then we come out with the innovation. Okay, we combine a lysine with rose, so it hopefully to reduce the smell of garlic. Okay, another. Okay, so actually this is my group of research here that actively involved in natural product or herbs plants. Okay, I'm heading my small group here. Okay, so I'm a biochemist in my field. Okay, so in our group, okay, there are microbiologists. Okay, Dr. Iza, okay, a microbiologist, and also Puan Nur Hayati. We also have oral biologist lecturers to help us to understand the nature of oral cavity. We have two lecturers. Their field are oral biology, and also we have also lecturer from a physiology department, Puan Bibi. Okay, this is my group. Okay, we call it natural approach to oral health, and I'm heading the group. Okay, so our niche area is about natural product, herbal medicine, oral cancer, and so on. Okay, so this is my student. Okay, they got the diamond when they are presenting the ficus diltodia. And this is also my colleague. Okay, my colleague when they are, I don't know, they are presenting what here, but they got something. This is only a final year project student. Okay, it's a dentist, bakal dentist, but they managed to got a very good result. Okay, this is me and my other two colleagues. This is physio lecturers. Okay, we are presenting our research finding in this conference, and we got best paper and also best presenter. I got both award, and my colleague got one by themselves. Okay, and this is me also with my students, my master student, and also my final year student. And this is how we bringing our product Okay, after we managed to do in the lab, okay, we try to do a formulations and a prototype. We bring to the conference, and we are in group presenting in the conference, and that is how we managed to got the awards. Okay, as you can see how apa this is kita meaning that I'm not alone in this project. I'm uh, I'm getting all the help from my colleagues. Okay, this is all my research colleague. Okay, with that, thank you. And this is some of the references that I use in this presentation. That's all for today.
Yes, thank you very much for the interesting lecture, Professor. I think your research group is wonderful as you have got a lot of awards regarding the research of herbal medicine in cancer. From the lecture, we can understand the importance of targeting the apoptosis pathway by triggering cell death but not causing inflammation. And I think this is very important as the principle of cancer medicine through several compounds that were mentioned. Okay, now let's move into the discussion section. I remind you again, if any of the participants want to give any question to Prof. Indah, you may write it in the chat box or directly ask it by raising hand. There have been several questions here. Let me mention one by one. From Dr. Lina, how can we determine active compounds from a plant while all compounds from the plant seem to be advantageous? Please answer the question, Professor Indah.
Okay, thank you, Dr. Lida, for the good questions. Okay, how can we determine the active compound from plant? Actually, before we embark to any research, we need to do a lot of reading. Okay, a lot of reading, and then how we start with MOD. Okay, we start with a reading first and search what are the others do, and then okay, we find the source and we find the expertise, and then we embark on this research. This is how we do our research. How we determine active compound? Basically, we manage to get help from the chemistry department. Okay, we have here, okay, a faculty of chemistry. They have a lot of compound, and if we collaborate with them, they can give us the compound. Meaning there is need a collaboration between the researchers.
Maybe we can collaborate more in the future, so
Yeah,
we can determine which active compounds can we do research together about this. Okay, the next question. Dr. Siti Hairunisa is raising hand. Maybe Dr. Nisa can ask the question directly.
Okay, thank you, Dr. Citra. Assalamualaikum, Prof. Indah. Congratulations, your project achievement. Thank you for your presentation. Very interesting topic about herbal medicine in apoptosis and cancer. When we talk about herbal medicine, then we also talk about secondary metabolic compounds. Some of the target of secondary metabolic compounds to inhibit the cancer cell by inhibiting the synthesis of purine, pyrimidine, etc. While we know that the synthesis of purine pyrimidine is also occurs in normal cell. So my question, how does compounds can recognize the mechanism of purine pyrimidine in cancer cell and in normal cell? Thank you, Prof.
Dr. Nisa, so how can we determine whether the herbal medicine attack the cancer cells but not normal cells? So can you please explain it, Professor?
In any setting of experiment, we need to have a control. We need to have a control positive and also control negative. Okay, when we want to see the effect on this different, so meaning that, okay, because I'm not really have time to discuss about the experimental design. Actually, we in every experiment setting, we also include the normal cell. Okay, when we treat our compound, we also treat the normal cells, and we want to see the effect. After we see that there's no effect to the normal cell, then we go to the downstream experiment. This is how we do it. First, we do the screening first, and we test also on the normal cells, and we see whether has an effect on the normal cells. And if you actually, I already mentioned in the conclusion, specifically about amodine, we already test actually this compound amodine on the non-transformed breast cells, and we didn't find any effect. And also it already been proved with other literature or research with other researchers also. They also see that our compound, that amodine, did not cause any harm to normal cells. If it is cause harm to normal cells, we will not continue the study. We immediately stop.
Is there any specific mechanism maybe only exists in cancer cells but not in normal cells, like a certain receptor like that?
Yes, say again.
Is there any specific target that is different between cancer cells and normal cells, so that the herbal medicine will affect cancer cells but not normal cells? Like, is there any specific receptor or specific pathway like that?
Because we are only focusing on the apoptosis. Okay, we are only focusing on apoptosis and not other markers like looking on the purines and pyrimidine contents. Actually, when you looking on the apoptosis, the assay is already complete. Okay, you can actually by using that assay, you know already that this this compound not harm to the normal cells. Okay, we not see to other markers. Okay, I'm not very familiar with other receptors or markers. I'm only in my research, I'm only focusing on this specific apoptosis marker, protein and gene expression of markers involved in apoptosis, both in intrinsic and extrinsic.
Okay, thank you very much, Professor. Is it clear, Dr. Anissa?
Yes, Dr. Chitra. But one more.
Okay,
maybe
please.
One more question.
Please.
Okay. Thank you. And based your research experience, Professor, maybe you know how the effectiveness of the mixture of various extract to inhibit the cancer cell. Is it more effective using mixed extract or using pure compounds?
Actually, okay, to prove that, okay, whether it mixed or pure compound, we need to do a very specific test, what we call synergistic test. When we do the synergistic test, okay, some compound better work alone, and some compounds better mix. In my study, I always mix my compound with the current treatment available. For example, with my study of amodine, I mix with tamoxifen, okay, the drug available to treat the breast cancer, and it shows the synergistic effect. And this is how we know it. Because we do the synergistic assay. There's certain assay to do that.
Thank you. I think it is interesting, and we can perform more research and even collaborative research involving this synergistic effect, right, Professor? Thank you for the answer. And The Lalisa Veterini also asks, the compound of allicin as anti-cancer, how can this compound target the cancer cells only? Will apoptosis also occur in normal cell? If the same to normal cells, then what are the advantages of allicin compared to chemotherapy treatment? Thank you, Professor.
Okay. Okay, right now, okay, so the lysine, we talk about the lysine is still undergoing study, okay. We see the anti-cancer properties of lysine, okay. How we will know that actually apoptosis also occur in normal cells, but it's okay because apoptosis is a natural way of cell suicide. It will not cause harm to adjacent cells. And this is actually good if the apoptosis happen. What we want to avoid is necrosis. And what are the advantages of lysine compared to treatment? Right now, okay, because right now we are but I'm not have time to explain my result. But right now, what we see about a lysine, when we compare with the current treatment cisplatin also kill the normal cells. We can see the features of necrosis happen in normal cells. But when we test the lysine on the normal cells, there's no toxics or less toxics to normal cells. So in cancer research, actually, we want to have the good drug that specifically attack the cancer cells, not the normal cells. This is the aim of the cancer research, actually. So this is what we are targeting, the objective of our experiment, actually. So every time we must involve the control or non-cancerous cells.
Okay, thank you for the answer, Professor. Dr. Lisa, is it clear? I think the apoptosis pathway itself is also very interesting as a target for cancer therapy. Okay, because the time is limited, even though we still have several questions, but I guess we should end here. Thank you very much, Professor Indah. We have learned from your lecture about several plants which have the potential for anti-ulcer and also anti-cancer. And also we have learned about their molecular medicine by targeting apoptotic cell death. This is very principal for drug development, including the application of herbal medicine. So it means we still have a lot of chance to study and perform research in this field, and maybe in the future. Some of our lecturer and also our students, especially from graduate and postgraduate study program, have also conducted research on herbal medicine. So I guess we could have a research collaboration together regarding this topic. Right, Professor?
Yeah, inshallah.
Inshallah. Okay, we hope we could still keep in touch with you and collaborate more in the future. By the way, Professor, could we copy your presentation as it is very insightful and important for us?
Yeah, sure, can.
Okay, thank you very much for the permission. We will copy and email it to the participants. So make sure all of the participants fill the data in the form completely, especially the email. Thank you very much.
Should I email the slide or just give the
Just give it to me, Professor. I will share it to the participants. Thank you very much. Thank you very much, Professor Indah, for giving us the lecture, even in the fasting month, Ramadan, and noon time. It's sleepy time, but the lecture is pretty interesting, and you are also full of spirit, so we can follow the lecture well. Okay. Maybe before we close the lecture, let us all take a picture. Maybe Mbak Lia can give comment to take the picture. Mbak Lia, tolong diinstruksikan. Please turn on all of your camera. Tolong dinyalakan semua kameranya. lihat bisa dipandu ya untuk fotonya.
1, 2, 3.
Sudah belum? Semua slide-nya sudah ya?
Yang lainnya belum.
Ya, semuanya.
Untuk yang berikutnya, ya. 1, 2, 3.
Sudah semua?
Satu slide lagi.
Ya.
1, 2, 3.
Sudah ya, Mbak?
Sudah, dokter.
Okay, thank you very much. Now I'll return the time to Dr. Emma as the MC for today.
Thank you, Dr. Chitra. Thank you very much, Prof. Indah, for very interesting lecture. And hopefully we can have many benefits from the lecture in the future. And thank you also for Dr. Chitra for moderating the discussion. And we would like to announce the certificate. We will send the certificate through email that is registered in attendance. So please kindly fill the attendance. Thank you very much. And hopefully we can see you soon next time in other scientific occasion. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
So can I leave?
Yes, Professor Indah, thank you very much.
Thank you very much. Assalamualaikum. Selamat berbuka.
Waalaikumsalam warahmatullahi wabarakatuh.
Lift dulu.
Ya, terima kasih semuanya.
Ya, izin lift juga ini, Bu.
Terima kasih, Pak.